Hi, good afternoon. Slides. Okay. Brilliant. Okay. So in the next 14 minutes, I'd like to tell you a little bit about our company, Cell Therapy Limited. If I could, perhaps I'll tell you a little bit about the story of how the company was founded and what our original hypothesis, scientific hypothesis, was back in 2009. So the company was founded by myself and Professor Sir Martin Evans. Martin won the Nobel Prize for his stem cell research in 2007. We believed in 2009 that tissue-specific, disease-modifying stem cells were there and needed to be discovered. We had, in all honesty, no evidence to support that assumption at all. It was just a hypothesis. Me personally, not being the scientist, backed Martin because he thought it was a great idea. And that's how we set up the company. We then funded the company ourselves in entirety until 2013, and then subsequently have done 2 rounds of funding and completed 2 phase 2 studies. This week, we were able to announce that we're progressing to an IPO. Yet to be decided whether it's the UK or NASDAQ, and that's on the back of potential, and I would say potential, and I'll explain to you why it's potential. Potential interest in conditional approval in Europe and in Japan, which would allow us to reach market in 2017. The US is a little bit behind that, probably in 2018. A little bit about the team. So Martin Evans is our Chief Scientific Officer, Founder and President. Martin works approximately 4 days a week in the company. We've located the lab approximately 5 minutes walk downhill from Martin's home, which is a great way to have the Nobel Laureate come into the office every day, which is a joy. And Martin does fundamental research. You know, in my humble opinion, he's still the most talented scientist in the field of discovering rare stem cells. And our underlying hypothesis, our underlying platform technology is that you can find multiple cell types that have tissue specificity. So far, we've been able to discover 19, and develop 3. And so Martin leads our discovery efforts. Dr. Sabina Sultan, who's on the right-hand side there, leads our efforts to translate those fundamental discoveries into preclinical and ultimately into clinical programs. We have a very illustrious board and scientific advisory board. Our company is chaired by Digby Lord Jones of Birmingham, who's a lawyer, and we have a number of eminent clinical collaborators. 2, which I think are particularly interesting, are Professor Steve Westerby, who's the Professor of Heart Surgery at the University of Oxford, and Professor Armin Keating, who is former President of the American Society of Hematology. As we move closer to commercializing in 2017, we've added a lot of people who have commercialization expertise, so people who have actually launched products across Europe and across the US. Very quickly, my background very quickly, I was a global head of emerging technologies at Roche, the big pharmaceutical company working at headquarters in Basel, essentially with the responsibility for looking for the next disruptive technology. Here's our pipeline. So apologies for the simplicity of the names, but given the science is quite complicated, we thought it would be sensible to have names that were quite descriptive. So HeartCell is one product. We have 3 indications. The lead indication is essentially a New York 4. That's how you would describe it in the United States. So heart failure is defined in grades, New York 1, New York 2, New York 3, and New York 4. New York 4 is the most severe type of heart failure. These patients have the most severe type of heart failure, New York 4. In Europe, the subclassification is different, and therefore you define these patients by an anatomical marker. So these patients have coronary artery malformation, which leads to them having a very poor prognosis. So typically these patients have somewhere between a 41 and an 89% mortality at 2 years. So approximately 70% mortality is what you see in this class of patients. Based on our very preliminary data, which I'll show you, in one study, the regulator asked us to also consider treating children. And the two indications that they asked us to look at, so this is the European Medicines Agency, asked us to look at Kawasaki's disease and bland white garland syndrome, extremely rare ultra-orphan drugs, except in Japan, where Kawasaki's has an incidence of about 148,000 patients per year. So that's one cell type. So that is one subtype of our, what we believe is a unique mesodermal progenitor cell, which is not an MSC, it's not an MPC, it's not anything else. It's a unique cell type, which is allogeneic, off the shelf, with cost of goods less than 10%. I think my colleague who spoke before, Raul, mentioned it. It's really important if you're developing a stem cell product that you have to be able to match the kind of economics that you would see from a typical biological product. So that's how we decided to progress our products. The second product in that cardiac franchise has a totally different phenotype, so a totally different type of cell, which has over 100 cell surface markers that are different to heart cell, and that's called myocardion. Whereas heart cell is delivered at coronary artery bypass, so open heart surgery, myocardion has different physical properties, and therefore that cell can be delivered through a catheter. Essentially, that cell has higher resistance to tensile stress and higher ability to resist hypoxia, which are the conditions that you see normally within a catheter. Tendon cell is a fundamentally different product. It's platelet derived, so a whole different cell type again. And I guess we're particularly proud of that because that has been able to show significant efficacy in a double-blind placebo-controlled study. So the sort of industry standard results that you would see from potentially a non-cellular product. Again, allogeneic, off the shelf, and perhaps what's interesting about that is that's a topical. So it's an acellular topical product that you use for tendon injury. And then skin cell, T cell, and the other programs are early programs. We in the company do not have, we don't disclose anything about products that haven't completed clinical trials. My timer isn't running, so I don't know. Well, I will go as quickly. 5 minutes left, or okay. I'll go on. Oh, that's great. My timer is now running. So heart cell fits what you would call in pharma a target product profile. So if you're in pharma, when you look at an opportunity, you try to put this opportunity into a framework that allows you to compare this product versus the other 200 products that you'll see every year. And that's this basis of this concept of a target product profile. And from that, you can derive the economics, you can look at the positioning, you can see whether this product would be first to market, best in class, etc. And so this is very simply categorizing. So it's an off-the-shelf, it's allogeneic, it's got less than 10% cost of goods, it's a single application at the point of cabbage, and it's developed wholly from both non-embryonic cells. It's xeno-free. We also haven't used any embryonic techniques. That's important in Europe because of the, let's call it unusual ruling that the European Patent Agency made against using any embryonic techniques for products if you want to patent them in Europe. They're both, so this product is an orphan indication with 2017 to 2018 launch dates. And as you probably know, orphan drugs confer a level of exclusivity either at 10 years or 7 years, depending on the market. Oh, that's actually, in the top right there, you actually see the cells under magnification, and then you see the cells ready to inject. We spent quite a lot of time making sure this product was exceptionally easy to use. So while we believe there's a high degree of science in developing the product, we do believe that there are lessons to be learned from products that had great efficacy in clinical trials, but then were not utilized. So this does not require any sophisticated processing by the surgeon or the cardiologist before use. It requires 1 hour in a water bath. And so we find that makes it very easy for the clinicians to use it. These are our clinical trial results. let me categorize this in exactly the same way as when I presented this data earlier this month at the European Society of Cell and Gene Therapy. All we can say from these results is it's a promising signal. The reason that's all we would claim is because this is a small study, 11 patients in an open label single center study. The reason it's only 11 patients in a single center open study is because when we applied for a double-blind placebo-controlled study, the ethics committee rejected that on the basis that this patient group has 70% one-year mortality. So what's intriguing, and why we believe perhaps that these results could be categorized as promising, is in a population with that high level of mortality, at 2 years we have 100% survival. So we expected to see a significant amount of mortality in this patient group, and now, on average, at 28.4 months, we have no mortality. What we do see, which is statistically significant, is an improvement in left ventricular ejection fraction, so heart function. These patients are terminally ill heart patients that had very poor heart function, and what we can see is, first of all, they're all alive. Secondly, that their heart function has improved significantly, 30%. And the reason we believe their heart function is improved is because for the first time, you're able to see scar reduction from an allogeneic product. So you're able to see meaningful scar reduction, 40%, which again is statistically significant on average across all patients. This translates into a significant quality of life improvement. The reason we did multiple endpoints there, just to give you perhaps a little bit of a thought process behind this, was very simply, at this point, when we started this study in 2012, we were still funding the company ourselves. And so frankly, we wanted to know whether we actually had something. So what we tried to do was to set up a study that we would like to see that would convince us that we might have a signal. And so we're absolutely delighted. It's beyond a surprise to us that all of our patients are still there, but obviously we're absolutely delighted to see that level of survival. Given time, I'm going to skip through this, but if I could just ask you to look perhaps at the images. This is just to put that survival data into context. So Nick was our very first patient. We treated Nick on the 28th of November 2012. Nick was 49 years old. He'd suffered 3 MIs, so 3 heart attacks, before we treated him. He was in New York Class 4, the worst type of heart failure. His heart function, his left ventricular ejection fraction, was down to 27%. The reason for that was because nearly half of his heart, 47% of his heart, was covered by scar. When you have a scar on your left ventricle, you lose both the contractile cells, but you also lose the elasticity of the pump. The heart is an elastic pump. So at a significant level of scarring, above 18% scarring, these patients have very significant mortality. And this is typically characterized by 70% mortality. And as I say, I would just simply say we're delighted that Nick and all the other patients remain alive and have seen significant improvement. Nick has gone from a 27% heart function to 45%. His scar has shrunk from 47% down to 22%. Very quickly, the images you see there, green is bad, red is good. Green shows non-vital, non-metabolic heart muscle, myocardium, and red shows active. What we were able to see with every patient, because we did serial imaging with every patient, was that at the site of injection, you saw contractile tissue, where there was no contractile tissue before. So perhaps, because it's an early signal, this is the first example of allogeneic heart regeneration. And we saw that uniformly across all of our patients. I am running out of time, so I'll skip forward very quickly. Our second product is called Tendon Cell. It fits our company's profile, so it's an allogeneic, off-the-shelf product with low cost of goods. It's topical in a 34-patient, double-blind, placebo-controlled study versus physiotherapy. So we took gold standard. What we saw was a 20% or a 23%, depending on the 2 endpoints, improvement above and beyond physiotherapy that was sustained for 6 months and for 3 months. That's intriguing because you don't normally see an improvement in this patient group. It's a large market, and we're progressing with partnerships on this product. HeartCell, we're progressing on our own. All of this technology is based on our ability to perhaps look at, with a higher level acuity, at the marker profile of these cells, which drives us to be able to define these cells very accurately. And on the basis of that, we look back at our hypothesis of 2009, and perhaps at this point, it's looking a little bit less crazy. Thank you very much for your time.